HESI A2 cell structure and function. Question 1. Impairment of which process would most directly prevent the following function? Moves small nonpolar molecules down a concentration gradient? Option A, internal respiration. Option B, atrial natriuretic response. Option C, simple diffusion. Option D, metabolic alkalosis. The correct answer is option C, simple diffusion. A key clue is that this process does not require a carrier protein or direct energy input. It also moves small nonpolar molecules down a concentration gradient. That combination is central to moving oxygen through a cell membrane. Question 2. Which option identifies the process most important for secreting neurotransmitters and hormones because it releases vesicle contents outside the cell? Option A, messenger RNA. Option B, exocytosis. Option C, placenta. Option D, transcription. The correct answer is option B, exocytosis. A key clue is that this process requires vesicle fusion with the plasma membrane. It also releases vesicle contents outside the cell. That combination is central to secreting neurotransmitters and hormones. Question 3. Which option identifies the mechanisms most important for forming genetically varied gametes because they randomly distributes maternal and paternal homologs into gametes? Option A, crossing over. Option B, respiratory pump. Option C, independent assortment. Option D, skeletal muscle pump. The correct answer is option C, independent assortment. A key clue is that these mechanisms increases variation during the first meiotic division. They also randomly distributes maternal and paternal homologs into gametes. That combination is central to forming genetically varied gametes. Stressed about your HESI A2? We can take HESI A2 for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 4. Which option identifies the solution that draws water out of a cell? Option A, hypertonic solution. Option B, the adrenal cortex. Option C, temporal bone. Option D, protein. The correct answer is option A, hypertonic solution. This solution draws water out of a cell. It causes cellular shrinkage as water leaves. These linked features explain the role in predicting osmotic movement across a membrane. Question 5. A review item describes a phase that positions sister chromatids for equal separation. Which option identifies it? Option A, G2 phase. Option B, metaphase. Option C, G1 phase. Option D, anaphase. The correct answer is option B, metaphase. For organizing chromosomes during mitosis, this phase aligns duplicated chromosomes at the cell equator. It can also be recognized because it positions sister chromatids for equal separation. The remaining choices have different primary functions or locations. Question 6. A review item describes a process that includes phagocytosis, pinocytosis, and receptor-mediated uptake. Which option identifies it? Option A, calcium homeostasis. Option B, receptor-mediated endocytosis. Option C, iron absorption. Option D, endocytosis. The correct answer is option D, endocytosis. For internalizing large particles or macromolecules, this process brings material into a cell by forming membrane-bound vesicles. It can also be recognized because it includes phagocytosis, pinocytosis, and receptor-mediated uptake. The remaining choices have different primary functions or locations. Question 7. Which option identifies the cell structure whose key function is, controls movement between the cell and its environment? Option A, cytoskeleton. Option B, nucleolus. Option C, plasma membrane. Option D, rough endoplasmic reticulum. The correct answer is option C, plasma membrane. This cell structure controls movement between the cell and its environment. It consists mainly of a phospholipid bilayer with embedded proteins. These linked features explain the role in maintaining selective permeability. Question 8. Which option identifies the process whose key function is, moves substances down a gradient through membrane proteins? Option A, facilitated diffusion. Option B, DNA replication. Option C, placenta. Option D, vascular spasm. The correct answer is option A, facilitated diffusion. This process moves substances down a gradient through membrane proteins. It does not directly require cellular energy. These linked features explain the role in transporting glucose through a carrier. Are you looking for HESI A2 actual questions? Visit our website pokerexams.com to access HESI A2 study guide with actual questions and answers. Scan the QR code or click the link in the description to get HESI A2 answer key today. Question 9. A student is reviewing a concept involved in taking up cholesterol- containing particles. Which option identifies the process with this function, internalizes specific molecules after they bind surface receptors? Option A, transfer RNA. Option B, menstruation. Option C, receptor-mediated endocytosis. Option D, pinocytosis. The correct answer is option C, receptor-mediated endocytosis. The role tested here involves taking up cholesterol- containing particles. This process internalizes specific molecules after they bind surface receptors. It can also be identified because it concentrates selected substances in coated pits. Question 10. Which option identifies the cell structure with this feature? It includes microfilaments, intermediate filaments, and microtubules. Option A, smooth endoplasmic reticulum. Option B, cytoskeleton. Option C, centrosome. Option D, a lysosome. The correct answer is option B, cytoskeleton. This cell structure participates in organizing the cell interior. It supports cell shape and intracellular movement, and it includes microfilaments, intermediate filaments, and microtubules. Those details distinguish the tested concept from related choices. Question 11. A learner connects the clues, produces most aerobic ATP, and contains inner membrane folds that increase the area for energy reactions. Which option best matches both clues? Option A, Golgi apparatus. Option B, nucleus. Option C, microvilli. Option D, mitochondrion. The correct answer is option D, mitochondrion. The function described is that this cell structure produces most aerobic ATP. A second identifying feature is that it contains inner membrane folds that increase the area for energy reactions. Together, the two clues explain the role in supporting cellular respiration. Question 12. Which option identifies the process most important for building a primary protein structure because it joins adjacent amino acids in a protein? Option A, homeostasis. Option B, respiratory alkalosis. Option C, peptide bond. Option D, hemostasis. The correct answer is option C, peptide bond. A key clue is that this process forms during ribosomal synthesis of a polypeptide. It also joins adjacent amino acids in a protein. That combination is central to building a primary protein structure. Question 13. Which option identifies the process most important for completing cell division because it divides the cytoplasm into two daughter cells? Option A, aldosterone response. Option B, follicular phase. Option C, primary active transport. Option D, cytokinesis. The correct answer is option D, cytokinesis. A key clue is that this process uses a cleavage furrow in typical animal cells. It also divides the cytoplasm into two daughter cells. That combination is central to completing cell division. Question 14. Which option identifies the mechanism most important for forming genetically varied gametes because it exchanges DNA between homologous chromosomes? Option A, crossing over. Option B, independent assortment. Option C, respiratory pump. Option D, skeletal muscle pump. The correct answer is option A, crossing over. A key clue is that this mechanism occurs during early meiosis and increases genetic diversity. It also exchanges DNA between homologous chromosomes. That combination is central to forming genetically varied gametes. Question 15. Which option identifies the process most important for absorbing nutrients across an epithelial membrane because it uses energy stored in one ion gradient to move another substance? Option A, receptor-mediated endocytosis. Option B, secondary active transport. Option C, transcription. Option D, fatty acid absorption. The correct answer is option B, secondary active transport. A key clue is that this process may operate as cotransport or countertransport. It also uses energy stored in one ion gradient to move another substance. That combination is central to absorbing nutrients across an epithelial membrane. Question 16. Which option identifies the process most important for sampling the surrounding fluid because it takes an extracellular fluid and dissolved solutes? Option A, pinocytosis. Option B, respiratory acidosis. Option C, calcium homeostasis. Option D, sodium-potassium pump. The correct answer is option A, pinocytosis. A key clue is that this process forms small vesicles without selecting a single target molecule. It also takes an extracellular fluid and dissolved solutes. That combination is central to sampling the surrounding fluid. Question 17. A physiology item gives two clues. It checks replicated DNA and prepares division machinery, and it occurs after DNA synthesis and before mitosis. Which option identifies the phase? Option A, prophase. Option B, G1 phase. Option C, anaphase. Option D, G2 phase. The correct answer is option D, G2 phase. The role tested here involves progressing through interphase. This phase checks replicated DNA and prepares division machinery. It can also be identified because it occurs after DNA synthesis and before mitosis. Question 18. A physiology item gives two clues. It synthesizes lipids and assists with detoxification, and it also stores calcium in specialized muscle cells. Which option identifies the cell structure? Option A, Golgi apparatus. Option B, a ribosome. Option C, secretory vesicle. Option D, smooth endoplasmic reticulum. The correct answer is option D, smooth endoplasmic reticulum. The role tested here involves supporting membrane production and chemical processing. This cell structure synthesizes lipids and assists with detoxification. It can also be identified because it also stores calcium in specialized muscle cells. Stressed about your HESI A2? We can take HESI A2 for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 19. A learner connects the clues allows cell growth, normal function, and DNA replication and includes G1, S, and G2 phases. Which option best matches both clues? Option A, G1 phase. Option B, interphase. Option C, S phase. Option D, anaphase. The correct answer is option B, interphase. The function described is that this phase allows cell growth, normal function, and DNA replication. A second identifying feature is that it includes G1, S, and G2 phases. Together, the two clues explain the role in preparing a cell for division. Question 20. A student is reviewing a concept involved in ending nuclear division. Which option identifies the phase with this function? Re- forms nuclear envelopes around separated chromosomes? Option A, prophase. Option B, G1 phase. Option C, telophase. Option D, anaphase. The correct answer is option C, telophase. The role tested here involves ending nuclear division. This phase Re- forms nuclear envelopes around separated chromosomes. It can also be identified because it allows chromosomes to begin decondensing. Question 21. Impairment of which cell structure would most directly prevent the following function? Links amino acids according to messenger RNA instructions? Option A, a ribosome. Option B, cytoskeleton. Option C, plasma membrane. Option D, centrosome. The correct answer is option A, ribosome. A key clue is that this cell structure may be free in the cytosol or attached to rough endoplasmic reticulum. It also links amino acids according to messenger RNA instructions. That combination is central to building proteins. Question 22. Which option identifies the process that moves 3 sodium ions out and 2 potassium ions in? Option A, set point. Option B, tubular reabsorption. Option C, oxygen diffusion. Option D, sodium-potassium pump. The correct answer is option D, sodium-potassium pump. This process moves 3 sodium ions out and 2 potassium ions in. It uses ADP and helps maintain the resting membrane potential. These linked features explain the role in preserving cellular ion gradients. Question 23. Which option identifies the process whose key function is, moves water across a selectively permeable membrane toward higher effective solute concentration? Option A, osmosis. Option B, respiratory compensation. Option C, cleavage. Option D, metabolic acidosis. The correct answer is option A, osmosis. This process moves water across a selectively permeable membrane toward higher effective solute concentration. It is driven by differences in water potential rather than ADP. These linked features explain the role in balancing water between fluid compartments. Question 24. Which option identifies the cell structure associated with allowing cell communication? In both clues, it binds a specific signaling molecule outside the cell, and it initiates intracellular responses without requiring the signal to enter. Option A, cell membrane receptor. Option B, Golgi apparatus. Option C, cilia. Option D, peroxisome. The correct answer is option A, cell membrane receptor. The function described is that this cell structure binds a specific signaling molecule outside the cell. A second identifying feature is that it initiates intracellular responses without requiring the signal to enter. Together, the two clues explain the role in allowing cell communication. Question 25. Which option identifies the process whose key function is engulfs large particles or microorganisms? Option A, metabolic acidosis. Option B, phagocytosis. Option C, endocytosis. Option D, secretory phase. The correct answer is option B, phagocytosis. This process engulfs large particles or microorganisms. It is especially important in certain immune cells. These linked features explain the role in removing bacteria and cellular debris.